Hello and welcome to From the Lab. I'm at the Paris Brain Institute, which is one of the world's leading organizations involved in research related to brain disorders. I'm here to meet Dr. Nicolas Renier, who was a part of an international study to determine the impact of SARS-CoV-2 on the human brain. Thank you, Dr. Renier, for joining us. First of all, can you tell me the nature of the study and what did you find out?
Yes, so this is an international study where we worked with partners at Yale University in the United States from the lab of Akiko Iwasaki. And this study is trying to address the question of whether the SARS-CoV-2 virus could infect neurons in the brain. And that study was based on early report that highlighted the possibility of brain damage in some of the early patients afflicted by this disease. So the lab of Akiko Iwasaki at Yale developed in vitro models to study the effect of the virus on neurons. And here at the Paris Brain Institute, we characterized mouse models and human patients infected by the virus, and to see whether the virus could infect the neurons in the brain.
What did your study find out?
So the biggest surprise we had at the beginning of the study was to see both in the in vitro model and in the animal models that the virus was actually infecting a lot the neurons in the brain. In vitro, our colleagues found that this infection would change the metabolism of the neuron, thus could affect the oxygen demands from the neurons, which could be an hypothesis that might explain the loss of neuron and neuronal death following viral infection. However, these part of the studies were done in vitro or in models. So the biggest question that we have now that we still need to answer is how bad the infection that we see in the lab can be on human patients. And so I want to be very clear that our study shows a possibility of the virus to affect in a serious way the neurons in the brain, but the study does not definitively say that this is actually what happens in the human patients, and more research is needed to address this point.
What does it mean when neurons get affected?
So when neurons get infected, they essentially become virus factories, and they start to churn out a lot of virus in their environment. And what was seen in this study is that as a result of producing the virus, the demand of those neurons for oxygen rises, which means that the neighboring neurons which are not infected by the virus actually do less well than the infected neurons because they get deprived from this oxygen from the infected neurons. And then we start to see in these models neuronal death in the neighboring regions that actually, in a surprising way, did receive less viral infection than the actual infected neurons.
What does neuronal death mean?
So neuronal death is pretty much permanent. So if you lose neurons in the adult brain, you don't recover them later. So it's pretty serious effect that can be seen normally in neurodegenerative disorders. But here, in those models, was seen following the viral infection. So the biggest question that we need to answer, and which we don't know the answer to yet, is whether the virus could have bad interactions between the cerebral infections that were seen here in this study and potentially other diseases that might be concomitant to the viral infections, such as Alzheimer's disease or Parkinson's disease, where the presence of the virus in the brain could interfere badly with the ongoing disorder.
Now, can you tell us what exactly did you do in this lab?
Yeah, so in this lab, we are experts in 3D imaging of the brain. So we were asked in this consortium for this study to determine the presence of the virus and its distribution in the brain using novel technologies that we have developed here at the Paris Brain Institute, which showed us exactly what are the identity of and the locations of those infected neurons. And then what we also did was a characterization of the vascular damage in the brain that can be caused by the presence of the virus. And using our novel technologies, we could show that the presence of the virus in the neurons was also affecting the vascular system in the brain, which could be a potential explanation as to how the presence of the virus could lead to vascular damage, which was seen in a few of the patients infected with the virus.
Now, most of the symptoms that are associated with COVID-19 are related to the respiratory system. Could mutations in the virus result in other organs getting infected?
So, even though SARS-CoV-2 was originally described as a respiratory virus, and for sure the respiratory tract is the first organ infected, now it seems that it is actually a systemic virus. So, in many patients, it's not only the brain, but you also have the skin that could be a target of the virus, or kidneys that could also be a target of the virus. So it is, even though most of the dramatic evolution of the disease is linked to the respiratory tract, many other organs in the body are affected by the virus. As to whether the mutations could lead to a differential targeting of the organs, in terms of viral mutation, anything is possible. I would say that this is maybe not the likeliest. The virus uses this door to enter into the cells, and this door, called ACE2, is rather ubiquitously expressed in the body. So we could say that the lock is already present in most of the organs in the body. So the virus has this natural capacity to infect already today most of the organs.
Thank you very much, Dr. Renier, for your time. That's all we have for now. See you again from a different lab.